Good morning, I'm Elise Randall, and here's the latest update from Spectrum Autism Research. Leo Kanner's 1943 paper on autism by Gerald D. Fischbach. Donald T. was not like other 5-year-old boys. Leo Kanner knew that the moment he read the 33-page letter from Donald's father that described the boy in obsessive detail as happiest when he was alone, drawing into a shell and living within himself, oblivious to everything around him. Donald had a mania for spinning toys, liked to shake his head from side to side and spin himself around in circles, and he had temper tantrums when his routine was disrupted. When Kanner met Donald, his suspicions were confirmed. In addition to the symptoms the letter described, Kanner noted Donald's explosive, seemingly irrelevant use of words. Donald referred to himself in the third person, repeated words and phrases spoken to him, and communicated his own desires by attributing them to others. Kanner described Donald and 10 other children in a 1943 paper entitled Autistic Disturbances of Affective Conduct. In this initial description of infantile autism, which went on to become a classic in the field of clinical psychiatry, Kanner described a distinct syndrome instead of previous depictions of such children as feeble-minded, retarded, moronic, idiotic, or schizoid. In the words of his contemporary Erwin Schrödinger, Kanner thought what nobody has yet thought about that which everybody sees. In recent years, experts have called attention to the inability of children with autism to understand that others have beliefs that are different than their own. These deficits prevent normal social intercourse, including the ability of the children to engage in joint fantasy or to empathize with the feelings of others. Kanner borrowed the term autism from Eugene Bleuler, who had coined it to describe the inward, self-absorbed aspects of schizophrenia in adults. But Kanner did not consider infantile autism an early form or prodrome of schizophrenia. The clinical signs were not identical, and unlike schizophrenia, Kanner's patients seemed to have autism from birth. Humane approach. Kanner was born in Austria and educated in Berlin. He came to the U.S. in 1924. In 1930, he moved to Johns Hopkins University, where he founded the first child psychiatry clinic in the country. Based largely on his clinical experience, he then wrote a textbook that defined the field of child psychiatry. His humanism is evident in his lifelong fight against the abuse of children with autism and intellectual disability, and his enduring concern for their families. He also made extraordinary efforts to help physicians and scientists escape from Nazi-controlled territories. The spectrum of clinical conditions labeled autism soon expanded beyond Kanner's first description. In 1944, 1 year after Kanner's paper, Hans Asperger described children that he also called autistic, but who seemed to have high nonverbal intelligence quotients and who used a large vocabulary appropriately. Confusion remains about the distinction between Asperger syndrome and high-functioning autism. It is remarkable that although Kanner and Asperger were born in Austria and educated in Germany, and both were astute clinicians focused on the same problems, they did not refer to each other. This may initially have been due to isolation during the war years, but it does not explain 35 years of silence. Clinical definitions of autism continue to evolve. The current Diagnostic and Statistical Manual of the American Psychiatric Association, or DSM-IV, includes autism in a broad category of pervasive developmental disorders. This spectrum blurs at the edges with disruptive behavior, communication disorders, and intellectual disability at one end, and with behaviors now thought to be normal at the other. Repeated revision and expansion of the diagnostic categories has probably contributed to the gradual increase in the reported prevalence of autism spectrum disorders that has been evident since the mid-1980s. Controversies over clinical definitions may only be resolved with the discovery of biomarkers, biochemical, anatomical, or physiological measures that are specific to one or more aspects of autism. Biomarkers are essential for an understanding of brain mechanisms underlying the various autisms and for the development of useful therapeutics. Innate disorder. Kanner appreciated the need for biomarkers from the beginning. He noted a tendency toward autistic behaviors in some families and described autism as an innate disorder. He foresaw the need for research into the genetics of autism at about the same time that DNA was first identified as the bearer of genetic information. Today, genetics offers the best hope for discovery of relevant biomarkers. It took courage to offer a hypothesis of innate determinants in 1943. The prevailing view of the day, based on Freudian psychology, had been that autism was due to poor parenting, with much of the blame placed on frigid mothers, accused of rejecting their children. Studies of identical twins have since made it clear that genetics is a powerful force in autism's etiology, but it is also clear that autism is not inherited in a simple Mendelian fashion. Many genes, perhaps as many as 50, may be involved, each one adding to the risk of clinically manifest autism. As these genes are uncovered, clinical descriptions will be further revised, and some may disappear entirely. We may not speak any longer of autism or schizophrenia or bipolar disorder. Rather, traits that share the same underlying brain mechanisms and that may fall into similar therapeutic categories may be emphasized. The relation between autism and other disorders will be debated on a different level. The search for genetic factors that enhance the risk of autism is now in high gear. Evidence for changes in DNA sequence, structural rearrangements of DNA including submicroscopic de novo copy number variants, and epigenetic modifications of DNA have all been reported in recent years. Success in this effort will lead to a clearer idea about mechanisms that disrupt normal brain development and underlie the onset of autism. Identification of such risk factors and how they might interact with environmental influences will only be the first step in this effort. Progress will speed up once the entire cast of characters is identified. Other measures, including quantitative functional anatomy, revealed through powerful new imaging techniques, and quantitative estimates of proteins and gene expression, will contribute to the search for biomarkers. The need for research into the biological basis of autism and of other neuropsychiatric disorders is urgent. In the midst of the current excitement, Kanner's paper is worth rereading. Not only is it the source of much of what we do today, but it can provide new clues for those prepared to receive them. Further attempts to resolve the neural basis of autism will rely heavily on this seminal work. That's all for today. Check back next Monday for more content from Spectrum Autism Research, or go to spectrumnews.org.